Hi, I am Mohammad Zaib from Department of Plant Pathology, University of Agriculture, Faisalabad. I will present before you occurrence of vector population in tissue culture potato varieties and resistant germplasm against potato leaf roll virus under natural feed conditions and its confirmation through ELISA. First of all, potato production. It is cultivated in the 100 countries of the world, and China is the biggest potato producer in the world. It is estimated that the total of all third potato in the world produced in the China and India. You can see here the pictures. The top producing countries are the China, India, Russia and Ukraine. China produce 25.4%, India 12%, Russia 8%, Ukraine 5.9%. Potato in Pakistan. In Pakistan, potato is ranked at 22 crop of in the productions, and regarding vegetable, it is ranked as a first. It is grown in the 8 different ecological zones, and in Pakistan, it is growing season are 3: winter, spring, and autumn. In Pakistan, 13 varieties are mostly prevalent, and most of the varieties and potatoes are produced in the Punjab, Pakistan, which is 86%. Here is a map. You can see that the contribution of each province in the Pakistan. You can see the Punjab contributed 6%, Balochistan 4.5%, NWFP 9%, and Sindh 0.5%. Next, my objectives. Determination of various incidence and vector population. Comparison of various incidence and vector population in comparison to tissue culture variety and normally grown varieties. Last one, screening of available germplasm lines varieties under natural feed condition against the major potato viruses. Material and methods. Preparation of MS media, treatments, sterilization, tissue culturing, incubation, ELISA test, vector population, and data recording. Next, meristem tissue culture. Protocols. First of all, apical parts of the plant are cutted, then plantlets are developed, and differentiation of tissues occurs. Plant growth media. Plant growth media, which is MS media, which was invented in 1962 by Moresh and Folk. It contains all possible hormones and nutrients, major nutrients and macronutrients. Major means macronutrients and micronutrients, and hormones are abscisic acid, cytokinin, gibberellins, auxins, ethylene. What is carnitine? It is firstly defined adenine type cytokinins, phytohormones, which induce cell division and oxidative damage, produce deoxyribose sugar in DNA. Here you can see a figure which shows its chemical formula. Media preparation. Sugar was dissolved in the water, then agarose gel added in the water by placing it on the heater. Micronutrients and macronutrients, vitamins, irons also added in the water. Mix these both mixture and make in 1000 ml. After preparation, different concentration of carnitine should be added. Why carnitine is be added later? Because it is unautoclavable. Treatments. Following 5 treatments were used while the culturing. Treatment 1, 0.5 ml per liter. Treatment 2, 1 ml per liter. Treatment 3, 1 ml per liter. Treatment 4, 1.5 ml per liter. Treatment 5, 2 ml per liter. Here you can see the figures performing the work in the lab. Meristem culturing. What is meristem culturing? Cutting the small portion of stem such as node, transfer the stem portion into test tube in the which contain MS media, covering it with the slip, tied it with the rubber band. Here you can see the diagram of the lab of incubation room. You can see you can compare these pictures which contain different concentration of carnitine. This table show when a concentration of carnitine will increased, then root length, shoot length, a number of nodes also increase at 1.5 ml per liter. But as we increase more concentration of carnitine, its shoot length, number of node, shoot and every weight increase, but other length, root length, shoot length and nodes also decreased. This was the scale that was used during the study. Sample loaded in the DAS ELISA. Following are the protocols used for the step ELISA. Coating the antibodies and coating the buffer in 200 microliter per well. Incubate at 4 centigrade or overnight. Then wash it 3 times. Then next load the sample and transfer to the sample. Add 200 microliter per well. Then incubate it at 4 centigrade or keep it at 24 centigrade at overnight. After that wash it 3 times and add conjugate buffer, then it will produce colors that can be seen in the next slide. This is slide, the sign showing yellow color show the negative control, positive control, and white color show the negative control. It is the table that it is the ELISA result. My result: screening against PLRV show that no variety was found to be highly resistant or resistance. 4 varieties show moderately resistance response, which are SL3064, SL721, SL174, SL1339, SL134 and SL114. 7 varieties show moderately susceptible response, which are SL52, SL1333, SL1343, SL933, SL143. Susceptible varieties were 12, which are SL1437, SL810, SL1150, SL720, SL1430, SL914, SL147, SL109, SL1022. Most important, which was the spread of vectors. Spread of virus is a mysis persica, which causes spreading of virus. You can see here. Next, screening against vectors. As we know, no variety was found to be immune or resistant. 4 varieties were moderate susceptibles, and we found 4 number of vectors per leaf. As we need to move to the moderately susceptible, number of vectors increased per leaf, which were 7. So in the susceptible were 12, and in the highly susceptible, which was desired variety used as a check, contained 22 vectors per leaf. DAS ELISA result: all the varieties show positive response except 2 varieties, SL1333 and SL174. Conclusion of my work: 6 lines found to be moderately resistant, 5 lines show susceptible response, and 9 varieties show most susceptible response. Mysis persica population was found less on the tissue culture screened varieties as compared to the commonly cultivated varieties. And 3 lines, SL1339, SL1333, SL1343, show negative response, while the others show positive response in the DAS ELISA test. Take home message: disease-free potato seed are being produced on a large scale with the help of tissue culture technique, having resistance both viruses and vectors. Screening results have a great impact in a variety development and breeding program for seed potato production. Thank you. Any question?
Okay, thank you.
One can use
कर लें,
इधर क्लिक करना।